So hi guys and welcome to Practical 2. So I hope you enjoyed Practical 1, but this one's going to be quite different. Instead of looking at protein evolution and using tools to identify proteins, we're now going to be looking at how mutations impact upon protein sequence and the organism that holds the genetic mutation. So we're going to be looking at a human protein, one of which is going to be a normal coding and one of which has a mutation. We'll go into that in just a second. I just want to say, obviously, once again, at the screen I'm looking at, the one that I'm talking over right now is obviously going to be different to the one that you're looking at. I don't have the same tabs as you because the website is still under development as I'm filming. Instead, the programs that I need are I'm gonna have them at the top of my browser here. They should eventually be part of the bar down here, but also you can still open them. There are instructions to do that on the page that we're going to go to now. So the worksheet here is going to give all the instructions. I would prefer if you watch the video the entire way through, and that's because having been a member of several bioinformatics classes and having taken some myself, things go wrong, and classes that I've taken do tend to find it far easier if I stand at the front and give examples so that we can work through together than they do if I leave them to work on their own off an instruction sheet. So let's begin looking at our protein. So bioinformatics can show us mutations in our sequences. The receptors CCR5 and CD4 are proteins normally found in the surface of T cells that form part of our immune system. When a person becomes infected with HIV, the virus attempts to dock onto the surface of the T cell using two receptors. After docking, it then infects the cell and replicates. Over time, the virus then destroys the cells. This destruction to the immune system over time can eventually lead to AIDS and death. However, there has been a mutation found in the CCR5 gene that makes people who are homozygous for this gene very unlikely to become infected with HIV. Now, I say very unlikely because I see some people say that it makes them completely immune. Now, I think there has been a couple of documented cases where people have got this mutation and got HIV, so I don't want to go ahead and say that they are completely immune, but it is incredibly unlikely. So those who are heterozygous for the CCR5 gene are more likely to become infected than those who are homozygous, but their disease progression is still much slower than those without the mutation. It is estimated that 1% of people who descended from Northern Europeans are homozygous for this gene, and a further 10 to 15% are heterozygous. Now, this mutation is called CCR5 delta 32, and we're going to be looking at why the gene has this name and what the mutation does to infer resistance to HIV in a person. So below is a representation of the normal CCR5 protein. You can see the protein in orange and the plasma membrane in grey and red. So I thought it would just be nice for you to see a visual representation of the protein that we're going to be working with. So we're going to take a look at the delta 32 mutation and compare it against a normal CCR5 receptor using bioinformatics tools. Just also as a side project, we'll also be using the PDP, Protein Data Bank, to take a look at the CCR5 protein that has been modelled. And that it won't be able to show us any differences between the mutated version and the normal version. But we will be able to visualize our protein. It will show how bioinformatics can visualize proteins and how we can kind of look at structures in more detail. So, like I've already said, I encourage you to watch the practical video the whole way through. So the first thing we're going to do is we're going to align our sequences, and that will help us show some differences. So if we highlight all of the sequence here, and we put it into Clustal. Now, those of you who have used Clustal Omega before, we're going to be using it in a slightly different way. So we've just pasted in the sequence, and what I need you to select is DNA. Now I know that the DNA sequence that we've got codes for a protein, but what that's actually asking you is whether your sequence is DNA sequence or protein sequence or RNA, and in our case it's DNA. So we're just going to go ahead and select DNA and press submit. So again, depending on where you are in the world and your internet connection and loads of other factors, this might be quicker or faster for some. Do feel free to pause the video. So it's going to become very immediately obvious to you once we scroll down why it's called a delta 32 mutation, and it's because of this here. So you can see all that this program has done. It has lined up our coding normal and our coding mutant line for line. Okay, so I can see no other problems with this, and this here is the delta 32 mutation because it's a deletion of 32 base pairs, so 32 nucleotides have been deleted at some point. So what we can now do is we can go to our worksheet, and it says when it is loaded, take a look at the lineup and can you see the difference, and I think we can safely say that we can, and make a note of what is different, and there is a deletion of 32 base pairs, so BP stands for base pairs, and that is why it's called a delta 32 mutation. Okay. So now we can see that there is actually a difference in the gene sequence. So let's take a look at what this difference means when the gene is translated into a protein. So we're going to be taking, what we're going to do is we're going to take the sequence, we're going to copy it again, and we're going to put it in a program called Emboss Transseq. So what this is going to do is this is going to take our DNA sequence that I've just pasted in there, and it's going to translate it into amino acid sequence for us. So you don't need to change anything. You just need to paste in the sequence and press submit. Again, this might take a while or it might not. Okay, so now this is showing up our coding normal and our mutant, and you can play around with this and you can show colours and things like that, but I don't think that's necessary. So what I'm going to do, I find it easier for me to show you what the difference is if I just pop it into a program like this, which is text edit. Okay, so we can see here we've got coding normal and we've got mutant. So we've got the protein starting with a methionine, which is great because that's obviously a start codon, and we've got that it's got a stop codon over here. It's got amino acid sequence in between. I can see no other stop codons, so this here is a stop codon. That's how they display it in this program. I can see no other stop codons. I can see no other problems. Now with this one here, we've got a methionine start, which is fantastic, but we don't have a stop here. Now those of you who are a little bit more eagle-eyed would have already spotted that the stop codon has actually nested itself part of the way into the sequence. Now obviously this means that anything after the stop codon does not get translated. So we might as well go ahead and we might as well delete anything after the first stop codon because it just will not get translated and it will not form part of the protein. So now we're looking at the protein sequence, that is actually translated into our protein that then gets folded up and becomes our working protein. So you can already begin to see the big difference here is that this one is significantly smaller. It is way smaller. And it has, obviously the 32 nucleotides have translated into an early stop codon. So if we scroll down to here, so down here is just a visual representation of everything I've just done on the screen. So you'll notice I've deleted some sequence after the first stop, and this is because after the stop codon is found by the ribosome, no other sequence is read, so it will not form part of the protein. So using our analysis, we can conclude that the mutation is called CCR5 delta 32 as it involves a deletion of 32 base pairs on the gene. So this causes a frame shift which results in a premature stop codon. So this premature stop codon causes translation to be stopped early and then results in a much smaller protein. This small protein cannot sit on the cell surface anymore and so doesn't properly function as a receptor, which means the HIV cannot bind to the surface of the cell and establish an infection. So obviously with this protein you might be interested to know why it comes from Europeans, why it's got European descent, and what kind of impact having a mutated protein has, other than obviously it infers a resistance to HIV, but does it have any negative impacts? Now I've done a little bit of research online about the CCR5 delta 32 mutation, and I found out why it's been descended in Europeans, like the current theory, and I've also had a look at what other negative impacts this mutated protein can have, because everything comes with an advantage and a disadvantage. If you want to have a look at that and you're really interested, then do feel free. I have to say it's really interesting. If you want to have a discussion about it on the forum, feel free. This does conclude our investigation into the CCR5 mutation, and I hope you found it interesting. We're now just going to take a look at what I said we would do, and we're going to have a look at the PDB. So if I just find that. So I'm just going to show you how bioinformatics tools can help us visualize proteins as well. So we've got the PDB ID here. And what we need to do is we need to go to the PDB website, so for MBS, and all we need is the front page, and all we're going to type in is for MBS, and we're just going to go ahead and search. And this website's great, by the way, guys. If you want to have a play around this website, it is fantastic. And here it is, a crystal structure of the CCR5 chemokine receptor. So you don't need to worry about, this is the paper that it's from, you don't need to worry about any of this, this is just how they got it. And what we can do now is we can kind of play around with it. So let's have a look here. Let's have a 3D view. So I know that my Mac struggles with this visualization. It might be that yours does too, or it doesn't. It's just that mine's a little bit slower than others. So here is the CCR5 chemokine receptor, and this is the non-mutated version. And this is the cool things we can do. So you can just kind of play around with it. You can zoom in, you can zoom out. You can put all kinds of things on it, so you can have it in cartoon, you can have it on backbone, you can have it on space fill. You can just kind of play around with the structure of it, and I just thought it might be really interesting for you is to have a look at how we visualize proteins using things like the protein data bank. You can have a look at the secondary structures, that's colored by the secondary structures, so the beta pleated sheets are a different color to alpha helixes. You can put all kinds of things in. We can go by sequence. We can have a look at symmetry. What's really interesting is hydrophobicity. which is obviously being afraid of water. And then you've got by amino acids, which is showing you all the different amino acids, which is kind of cool. Surfaces, you can also play with that. You can put your H bonds in. This is just really, this isn't part of the assessment and doesn't form part of the final quiz. I just thought it might be really nice for you to have a look. Another thing that you can do is you can hover over different parts and it tells you what amino acid it is, which I thought was really interesting. So this green here is, you got that one, isoleucine, valine. So if you scroll over, it'll tell you what amino acid it is as you go along. I just thought it'd be really nice for you to have a look and play around with proteins. If you don't have to stick with the CCR5 protein, I just thought it'd be relevant to our practical. You can use other visualizations, so it links to other visualizations as well, so you can visualize it. There was another one other than, there we go, selective viewer. So there are other viewers. I'm just going to stick with the simplest one. If you guys want to play around and you want to look in more detail, then do feel free, by all means. You can scroll forwards, you can have a look at the front, you can move it around for you. Subunit. So this is just really for you to play around with. If you want to go around, they've got some really great things if you want to visualize things. Oops. So they've got more visualizers, including human gene view and things like that. They've got analyzers, they won't necessarily need that. There's also PDV 101, which is where we learn things. It can tell you about molecules of the month, browsing all resources, etc, etc. So if you guys have really enjoyed that and you want to keep playing around with it, then do feel free. They do some really great things. So currently, the month that we're in is May, and the molecule of the month is Zika virus, which has obviously been in the news and not recently. So if you do find this tool real interesting, then do feel free to have a browse and a play, and you can visualize and analyze, and there is loads of things where you can learn. I have to say their educational resources are fantastic. So I hope you have enjoyed all of our practical work. If you have been stuck with anything's unclear, you think you found a mistake, I'm only human, mistakes do happen, then please do post on the forum or contact me directly, and I can try my best to help you fix it. So thank you very much, guys. I really hope you enjoyed it.